All right. This is a science talk, so please block the exits. Keep people from escaping, and we'll see where we end up. My talk is about evolution, and lots of things have been said about evolution, lots of things have been done. I want to make one disclaimer. I've only been given 2.5 hours to talk about this by the organizers, so I have to give a short introduction to some aspects to a summary of evolution. I'm going to skip over things, going to simplify things, and you're going to have to live with it. But the points I'm making hopefully will make some sense. Evolution, everybody knows the theory or thinks that they know it. It's a work in progress. That's very important. There's many things we understand. There's many facts that tell us evolution is right. There's not a single scientist that really works by scientific methods, that looks at facts and deduces theories that doubts the theory of evolution. That does not mean that the theory of evolution is there and it's not changing. We always discover more and we need to adapt our theory. It's very important. Some people think that because we sometimes discover something and we need to make slight changes to our theory, that the theory is not valid, and instead they come up with a theory for which there is no proof at all, and they think that's a much better option. I don't think so. All right. Let's start with this guy. As you can tell, very fashionable, French, right? JeanBaptiste Lamarck. He was one of the first people to come up with a coherent theory of evolution. He's done many more things, but his theory is quite extraordinary, and one of the aspects of the theory is that he believed in the inheritance of acquired characteristics. What does he mean by that is that, well, look at this giraffe. It's a very easy way to explain this idea by Lamarck. Everybody knows that giraffe has kind of a remarkably long neck. How does it get the long neck? Well, it's trying to eat leaves on the tree, and it stretches its neck. And therefore, the little kiddie giraffes will have slightly longer necks. And again, this repeats, and that's how the giraffe got a long neck. Seems a bit silly to us, but it's actually a great idea. He was going with the data that he had. Wonderful theory, except it's not right. In came Darwin, and enough has been said and done about Darwin over the last year especially. He's been great. One of the things that he did is he introduced it to two key concepts, namely variation and selection. And with variation, he just said, well, these giraffes, they don't stretch their neck. Well, maybe they do, but they are born with short and longer necks. There's just this natural variation amongst giraffes. And the lucky ones that have the long necks can reach more leaves. And as you know, you only think about sex after you're not hungry anymore. So they're going to reproduce because they're not hungry anymore. They're going to get little giraffes with slightly longer necks, and that's how evolution happens. So this is the selection part, and then there's natural variation. He didn't really say how the natural variation occurred. He didn't really have answers to that. He thought about it a lot. But he really separated the two processes. And this is also what made him so controversial, because it was very cruel. It's a very cruel way of having evolution. There's giraffes dying. There's poor giraffes with short necks dying here. All right. And this fellow here looks very stern. He's German. August Weismann, great biologist. And he really... One of the things he did is really try to prove that variation and selection are completely independent. And one way he did this, so he really sort of tried to kill the old idea by Lamarck that the length of the neck of the giraffe has really nothing to do with what it did in its lifetime. It's it's stretching out for trees. So one of the things he did, he's really very famous for this experiment, although it's not his best experiment. He took mice, as soon as they were born, cut off the tail, then bred more mice. As soon as the little mice were born, cut off the tail again, and just repeated this. And in the end, what he noted was that all these new mice, these little mice, even after he's done this for 30 generations, still had tails that were just as long as the original mice. So it's a great way of disproving Lamarck. I would say he should have relaxed, sat back, thought about looking at the Jewish male population, and he wouldn't even have had to do his experiment. So he came up with I think, a much more important finding, though, and really amazing work that he did, where he actually said that even early in our lives, and I'm talking about when we're embryos, what he called our germ cells, these are the cells that we will use to reproduce, are separated from the rest of the embryo. And you can see them here as little dots. And they separate, and we all know where in the end they end up. And the strong point in that, he was totally right about this, and the strong point about this is that pretty much what he was saying is when the giraffe stretches its neck, it's not stretching its testicles. So how can this have an effect on your germ cells? It's a very strong point, and again, at least for complex organisms, he set apart variation and selection. The forces that will select you are independent of this variation that you have. Again, a bit later, these two fine gentlemen here, Luria and Delbruck, were working in Cold Spring Harbor in the United States, where they were doing multiple amazing experiments. And one of them got them the Nobel Prize. And they were working on this virus here, which looks a bit like a moonlander, but it's a little bit smaller than it. It's called a bacteriophage. And this is good news for all of you. All of you non-scientists might not realize that these bacteria that make us sick, they actually also get sick. They also have virus infections. The only organisms that get away without being sick are the viruses themselves. But so bacteria do get virus infections, and they actually die from it. And that's what these guys were studying. And they also wanted to look at this idea, is variation independent of selection? And they came up with a very smart experiment. What they did is they said, well, let's start from one bacterial cell. And let's give it a lot of food so it will make lots of little bacteria, and they always divide. As you know, bacteria, they grow or they multiply by just dividing themselves into two. And they make clones of themselves, genetically identical, and so this is what happens. And they said, so these are the bacteria here, always dividing, and they said, well, we're going to at some point introduce a virus, and we're going to see what happens, because they've noted that when you introduce a virus to lots of bacteria, there's always a few bacteria that manage to survive. They are genetically, because their little children bacteria are also surviving, so it's clearly a genetic trait. Something has happened to their DNA, to their genetic material. And so something has happened. Some of these bacteria are resistant. And now the question is, this variation, because that's what it is, does it occur before the bacteria are ever in contact with the virus? Or is it when we infect this culture, these hundreds of millions of cells, that suddenly a few manage to become resistant? It's a very interesting question. And they were very smart. They said, well, suppose there is a mechanism by which when you infect the virus when you infect the bacterium with virus, it tries to become resistant some way. There's a mechanism. Then if you do this to 100 million cells, and you do it a few times to 100 million cells, you sort of expect that a similar number of bacteria will always become resistant, that manage to get there, right? The lucky few. Whereas, suppose that some bacteria become resistant while they're multiplying, here the blue dots here, you can have vastly different numbers when you repeat this experiment, because what can happen is, here you have a bacterium that becomes resistant to the virus very late in the reproduction, and there's only one in this whole population that's resistant, that's not killed by the virus. Here, though, you have what is called a jackpot event, and the name comes exactly from what you think it comes from. Very early in the reproduction of this first cell here, one of the two kids becomes, or maybe the parent, becomes resistant, and it starts dividing, and now half of your culture, but we're talking millions of cells here, are resistant. So you have this huge variation. So they've done the experiment, and what they found was this. And so they concluded clearly, and they've shown this mathematically, clearly some bacteria in this population were resistant to a virus that they've never seen before. And again, variation must be independent of selection. Now, I would claim, and other people have claimed, that their experiment does contain a pretty serious flaw. And I'm not saying they didn't deserve the Nobel Prize at all. They definitely deserved it. But one problem with their experiment is clearly that, well, they introduce a deadly virus. Maybe the bacteria have a mechanism to develop resistance or tolerance to this virus, but not to one that kills them instantaneously. Maybe they should have used some milder stress, some milder selection. So that's a problem there. And then, of course, later, after Watson and Crick and Rosalind Franklin here discovered the structure of DNA, and the whole molecular research started taking off, we sort of put everything together of the evolution theory into what is called the new synthesis. And that's sort of our current theory of evolution, where you have changes in the DNA code that are more or less random, they're independent of selection, and they give you differences. That's what's causing all these differences between us, and that's why some of us cannot get AIDS and most of us can, which is true, by the way. And so this is pretty much our theory. Now, I don't want to end here. What we've seen is that more and more evidence is emerging that the story is a bit more complex, and maybe variation and selection are not so completely independent as some people believed. And I got to know about this while studying this year. I did my PhD in a beer brewing lab. You know, it's one of the better places to start your research as a student. And I was studying yeast cells. Yeast cells, great genetic model organism, by the way. So actually one of the frustrations I have is, you know, try to be taken seriously by the people who need to fund you, or at a conference when you're working on beer and you go like, trust me, I'm doing real serious genetic experiments here. All right, so one of the things I was studying is yeast cells that are clumping together. It's called flocculation. So what you see here is a bunch of yeast cells that stick to each other and they settle out in this culture here. This is important for beer because it happens at the end of fermentation. This is what pretty much makes the difference between a clear beer that doesn't have any yeast cells in it and what we call a wit beer or a Weissen beer that has the yeast cells still floating around in it. And we were trying to find the genetics of this. What we found is this one gene here, FLO1, which stands for flocculation 1. It's a gene, and what is so special about this gene is that it contains a middle part that is extremely unstable. So this gene is, of course, made of DNA, like any gene. And the middle part of the DNA is extremely unstable. It changes much more than any other DNA. And what it is in particular is that it contains these things which are called tandem repeats. It's pretty much a piece of DNA that's repeated all time and time again. It's much longer than what you see here, but you get the basic idea. And what makes it unstable is that the number of these repeats changes very quickly. Every time the DNA is copied, you have a pretty high chance that the number will be different from what it was. This has been known for a long time, except people don't really expect it too much within genes. Usually you find these tandem repeats outside of genes, but here and in some other genes you find this. So what you have here is a piece of DNA or a particular gene that's changing more rapidly than other genes, and in this case it means that flocculation is changing, so this characteristic of the yeast, this specific thing compared to a long neck, if you will, is changing more rapidly than some other properties of the yeast. Now if you think that this is well, this doesn't matter. If you think that this is specific for yeast cells, you're wrong. Pretty much around the same time we were publishing our story, there's a great story published about dogs, and I don't know if you thought about this, but dogs are some of the most variable creatures on the face of the earth, especially when it comes to their shape. Just look at, you know, this Chihuahua and this Saint Bernard here. They're the same species in principle, and I say in principle. These things can breed. You just hope that the Chihuahua is not a female. Now, these are bred by humans, right? We have made these dogs by selection and whatever, but we didn't even use so much time for it. In evolutionary terms, these things are new. They are brand new, and they were they developed in a very short time. And one of the things that was found is that one of the key regulators that regulates, and I'm again talking about the gene, it's a regulating gene, and it regulates the shape of the skull, basically the shape of the dog as well. And it also has these unstable tandem repeats in it. And what these researchers found is that there's a nice correlation between how many repeats you have in this gene and how curved your snout is, or how long your snout is. And they also found that some other changes in another regulatory gene give you a sixth finger, sort of this little extra thumb here. And I didn't know this, but this is a characteristic of a specific breed of Great Dane dogs. And that's why actually this happened. It sort of happened in a very short time, and people now see this sixth claw, if you will, as a characteristic. So clearly it's not just happening in yeast, and there's more. One of the other things that people have known for a while, and we're also researching, is that the end of chromosomes are basically packages of DNA, that's how our DNA sits in the cell. Well, the ends of chromosomes here, the very ends, they change much more quickly. There's higher mutation rates, the DNA is not as stable, and so the genes that lie there, again, evolve. And if you're wondering, in humans, which genes are lying there, it's the genes that, for example, the genes that make us smell. And of course we have to recognize lots of different smells, and these genes are copying themselves and they're changing very quickly. In plants, a whole different mechanism, and this is a bit more complicated, I will try to fly through it. There's this particular protein, and this is a bit like your mother. This protein is the mother of the cell. It sort of checks all the other little proteins and it goes like, are you okay? You don't look very good. Here's your code. You should behave this way. Don't behave that way. It's sort of like a teacher mother. And so the protein really takes care that even if there's small mutations, changes in other proteins, that they still behave right. And if they don't behave right, they get degraded. Now what you see is that in times of stress, and plants also have stress, stress is a biological word for selection. It means you're not adapted to a condition. It means that you feel the burden of evolution pressing down on you, pretty much. So in times of stress, this protein, this function of this mother protein gets dialed down a little bit, and suddenly these plants start misbehaving. You know, they become weird, and that's because some mutations that you previously couldn't see now suddenly emerge. And although it's not proven, it seems like a likely theory that maybe this could serve as a mechanism to try and escape the stress. Because suddenly it's good to try and be different from what your mother was. And so maybe a few of these plants are better at surviving the stress and they will cope, and then maybe this mutation can get fixed and this and that. Another example comes from bacteria, and I'm just skimming over it again, but bacteria, in times of stress, again, they activate, and this is just to impress you, it's not very important. In times of stress, what they do is they activate a different protein to copy their DNA. And of course, a protein to copy your DNA is a very important protein because it shouldn't make too many mistakes, because that's how you get changes in DNA. And that's how you get natural variation, so you need a little bit of them, but you don't want too much of them because mostly your variation is not good. It wouldn't be so great if a giraffe got a neck that was 3 times as long because the heart couldn't cope with it. But in times of stress, again, it's obviously sometimes you need to choose between dying or gambling, and bacteria may be gambling. They activate this gene that's very sloppy. And so the DNA gets copied, but it has lots more changes in it. And maybe, although it's hard to prove, but maybe this is a strategy of bacteria to try and beat deselection, the evolutionary pressure that's pushing down on them. An even nicer example, I think, is the water flea here. Again, it's pretty mysterious still, but water fleas, swimming around, beautiful organisms, they have predators. And when they get, there's a family of water fleas and that gets eaten, there's some chemicals released in the water, and it induces the formation of the stickle here, which is called a spina. And the stickle makes the water flea a little bit less attractive for predators. Now that's all great, that's not so special. A chemical induces some morphological change. The weird thing is that the kids of this water flea will also have the spina. Even if they've never seen a predator, even if you take all the predators away, they will still have this for quite a while, for a few generations. So this comes very close to Lamarck, right? There's something happening in the course of the life of this organism. It changes something, and it's giving that information to its kids. It's getting pretty close to Lamarck. So this is the conclusion of the talk, and this is important. Does this mean that our theory of evolution really needs a major overhaul? I would say not at all. And people have often misunderstood, I guess, the things that I've said and published, and it happened recently in this Flemish or Dutch magazine here, where I wrote a piece telling you this, or writing about the same things that I'm telling you now. And this is the cover they came up with. I wasn't so happy because it looks like I'm sort of sawing the ground under Darwin. No. Here, this is what Darwin wrote literally about variation and selection. He says, I've spoken as if this natural variation was totally random in my book. Like it was just pure chance. But of course, I didn't mean to imply that. It just means that I don't really know what's happening, and maybe there's mechanism, it's much more complex. So Darwin was extremely clever. He thought about his theory. He knew exactly where the holes were and where he shouldn't really speak for one possibility or the other. So he actually incorporated it. It's only later that maybe we've gone a bit too far away from Lamarck. He didn't really dislike Lamarck's theory that much. Although, this is not to say that Lamarck's theory was right. I mean, I still think that it's mostly random, but there's some small changes here and there that make it a little bit less random than completely random. So what I'm saying is that through evolution, mechanisms have developed that make evolution not a complete chance. And then you might start wondering, how can this be, right? And there I would argue this just happens through the process of evolution. Suppose a gene becomes very unstable, and it's a housekeeping gene. It's a gene that doesn't need to change, or it doesn't need to change as quickly, or when it changes, it's mostly detrimental. Now, if such a gene becomes unstable, it's going to be a huge disadvantage for the organism that has it, and so it will be selected away. However, if a gene, for example, a gene that makes your skull a bit more flexible, like in the giraffe, and maybe you can, you get more giraffes with longer necks. If such a gene arises by pure chance, and this is pure chance, maybe it becomes an advantage for the organism and it stays there. It stays unstable like it was, and maybe that's how these things have evolved. Now again, like I said, our work sometimes gets misinterpreted, and sometimes it's quite funny, especially when it's the people that believe in creationism or intelligent design that take our work. This was the more funny things. This is a website called Uncommon Descent, and if you think about it, the title says it all. These people don't believe in the common descent. That's, of course, at the core of our evolution theory. And so we published a paper, a colleague in the US, and when I was still working in the US and I. And we were, again, in this paper, it's a more in-depth discussion of what I'm telling you now. And we were aware of the fact that some people might misinterpret this. So in the abstract, in the summary of the paper, which is pretty much the thing that everybody will read, we wrote specifically that our ideas do not go against Darwin. And then these guys here read the article, still wanted to use it for their ideas, and they said, well, to publish this into a reputed scientific journal, the authors needed to write something that their ideas do not go against Darwin, but they don't mean it. It's just a secret handshake to get into this good journal. So that's the secret handshake here. So luckily there was, then it becomes quite funny because there's reactions on this forum of people, and well, I can barely read it myself, but I'll try. So this is one of the people reacting to this, and he says, they quote some of the parts that we write in the paper, and it says like, error-prone DNA copying enzymes produce bursts of variability in times of stress. These mechanisms seem to tune the variability of a given characteristic to match the variability of the selection. That's something that we wrote. And it says, gee, it almost seems like a built-in response mechanism. Who'd have thunk? Darwin is so dead. That's what she writes. Anyway, so there's some people who did not misunderstand our paper and they reacted to this, and it's also fun to read this discussion because then the intelligent design people get on. It's all one great family, and it's kind of fun to have this. I really like this discussion, so I have nothing against people who come up with different theories. They're just wrong, but it makes it fun to discuss with them. All right, this brings me to the acknowledgements. And I have to acknowledge pretty much all these people here, which are the people that are doing all the hard work in my lab. Probably as we're speaking now, they're getting more results, so I can give another great talk and be the hero for this audience. They are chained to their benches. I have to remember to feed them tonight. But yeah, they really are the heroes of the lab, and there's many more of them. Of course, our group is definitely not the only ones doing this work. For people who are scientists and want to know more about these are some of the publications. This is the major one where we really sort of discuss all these things. There's more information on the website, and this is very important as well. These are the people that are paying us. Well, not me, but more the research. Thanks.